Hi, let's talk about the chemistry of life. Now, you may be wondering why we're going to talk about chemistry again. I already took the class, and this is a human body class, so let's talk about the body. But if you think about it, everything that you're made out of is matter, and the basic unit of all matter is the atom. So it really comes down to the chemical level when we talk about even large things like the human body. And secondly, when you talk about the functioning or the physiology of the human body, you need to look at the chemical processes that happen in order to make the different parts of your body work. For example, how does calcium, which is one of the elements, how does it actually help and allow for bone growth? How does your digestive system actually absorb the nutrients it needs to maintain the energy that you need to have? And then when you look at your bones and or not your bones, but your nerves and your muscles, how do they communicate with one another? It all comes down to the chemistry. So it's important that we actually review the stuff and make sure you have a good grasp and foundation and refresh your memory in case you forgot some of the things. Okay. So let's look at this periodic table. Everything that's highlighted in purple or green are pretty much the atoms or the elements that are found in living things, particularly you. And so we need to actually review the atomic structure and ions and isotopes and the different types of bonds. So let's look at this. When we look at atomic structure, hopefully you remember that this is actually the Bohrs Model, which you probably learned from chemistry, or the planetary model. It's not very accurate because of the fact that electrons don't behave in an orbit like the planets do around the sun. They're constantly in motion and bopping around from different orbit to different orbit, and it's hard to predict where they actually are. However, in chemistry, in order to learn it and teach it, we always use the Bohrs Model just because it makes life a lot simpler. Looking over here, we have the atomic number. The atomic number is the number of protons. Remember that we have 3 subatomic particles: protons, which are positive, electrons, which are negative, and neutrons, which are neutral and do not have a charge. And so this top number of the periodic table tells us how many protons, but indirectly it also tells us the amount of electrons, because atoms are electrically neutral. That means that their negative electrons will always equal their positive protons. Negative and positive will always cancel out. So if sodium has 11 protons, it also has 11 electrons and is neutral. The bottom number over here is its atomic mass, which is the atomic mass is always what is the nucleus composed of, and it's composed of protons and neutrons. And so because there are 11 protons, if we subtract 11 from 23, it will tell us that the number of neutrons is 12, and they make up the mass. They're the ones that have a significant mass. Electrons do have a mass, but it's so insignificant that we kind of say it's negligible, and so we kind of ignore the fact that electrons have a mass, even though they really do. The next thing I want to talk about is a symbol. The symbol is right here. It's usually 1 letter or 2. In a few it's 3. But you should know by now that when you write a chemical symbol or name, you capitalize the first letter always, and you lowercase the second or the third. Students get into a lot of trouble because when they write carbon dioxide, here's carbon dioxide. And we talk about carbon dioxide a lot in biology, but then I have biology students who write carbon dioxide like this, but that's cobalt. That's not carbon dioxide. So you could get into some trouble, so make sure that you write it correctly, capital or lowercase. The next thing I want to show you is this. This is the isotopic symbol. It tells us any isotopes, or really what the mass of the atom is. The mass number goes on top, whereas the atomic number goes on the bottom instead, as superscripts and subscripts in front of the symbol. Other things that I want to refresh your memory on, we're going to use the rule of octet, or the rule of 8s, which means that the first orbit contains 2 electrons, and 2 electrons only, and the most. Every orbit after that can contain a maximum of 8 electrons. You probably learned in chemistry that that's a bit of a lie when it comes to certain other atoms, but for our purposes in biology, we're going to still use the rule of octets. The very last orbit over here, this is called the valence shell, and any electrons in the valence shell are called your valence electrons, and those determine the reactivity and what it's going to react with and how it's going to react with other atoms. So keep that in mind. With the valence electrons, you could actually, the periodic table is set up so you immediately know how many valence electrons is in each element. So we have these different columns, which are called groups. So the first group is in the first group because it contains one valence electron. The second group contains two valence electrons. Third, well, we skipped the transition metals right here, but group 13 or group 3, sometimes called group 3, has 3 valence electrons. We have 4 valence electrons, 5, 6, 7, and all the way up to the most, which is 8. And we call this last column the noble gases because they are inert and they do not like to react with any other atoms, whereas these other guys usually do. These dots, remember the Lewis dot structure? We use 1 dot for every valence electron that it has. And so it goes from 1 all the way up to 8 different valence electrons around. And that's very important, especially when we talk about ionic bonds. So let's talk about ions first. An ion is an atom that has gained or lost electrons. So now it has a positive or negative charge. Atoms are neutral unless they gain or lose electrons, which you're always looking to do unless you're a noble gas. They're not stable unless they have a full valence shell of electrons. So if we look at this atom over here, which is lithium, lithium has 3 electrons but only 1 valence electrons, which means it does not have a full valence shell. So it's not stable. It's looking to get rid of that 1 electron or gain 7 more, but it's easier to get rid of that 1. So it will lose it to another atom, and when it does that, it goes from 3 protons and 3 electrons to 3 protons and only 2 electrons. And so that means that you have a positive 1 charge. Any positive ion is called a cation. So a cation has lost negativity, their negative electrons, which made them positive. Over here we have anions. Anions have gained electrons, and electrons are negative, so they gain negativity, so they become negative ions. And so in this case we have fluorine, which has 7 valence electrons, and it's not stable. It's looking for 1 more to complete its valence shell. It will take on another one donated from another atom that was giving them up, and once it has a extra electron, then fluorine has 7, 8, 9 protons. And it originally had 9 electrons, but now it has 10 electrons, which means it has a negative 1 charge, and that's called an anion. And so a lot of atoms form this way, and so you have sodium and chlorine. Sodium will give up its valence electron to chlorine, which is looking for 1, and because sodium becomes positively charged and chlorine becomes negatively charged, opposites attract, and so we get sodium chloride, which is table salt, through an ionic bond, which we're going to get to. Because the valence electrons determine how an atom will ionize, we could also use the groups to figure out how all the atoms will ionize when they form ions. So the first group over here, they all have 1 valence electron, which means it's easier for them to lose that 1 valence electron to become stable. So when they lose 1 negative, they're going to become positive. When the second group, it'll be easier to lose these 2 electrons to become stable. And when they lose 2 electrons, they now have 2 less negatives, which means that they're going to have 2 more protons, which makes them a positive 2. The third column, they will lose 3, so they will be a positive 3 charge. And then they could gain or lose the 4. Usually this column does not. They will form covalent bonds instead of ions, but it can happen. Now over here, this is where we change over. There's 5 valence electrons, which means it's easier to actually gain 3 more to become stable at 8. So when you gain 3 negative electrons, you become negative and get a negative 3 charge. And then here's a negative 4 charge and negative 5 charge. And, oops, I'm going the wrong way. Oops. Forget that. They will gain 2 more. They have 6, so they need 2 more negative electrons, and they need 1 more negative electron there. My bad. And the last column are the noble gases, and they're happy with what they have. They have full valence shells, which is why they're noble and why they're inert and why they do not react with other elements. Let's look at the importance of ions in the body. So why do we even care about ions? Who cares about this? But it's actually how your cells communicate with one another. It's how your muscles and it's how your nerve cells will actually conduct electrical impulses that send signals to your brain and back into all the different cells in your body. So they're super important, and you have to maintain a certain level of ions across your cell membrane. And if we look at this picture over here, sodium, here's a sodium ion, so there's a lot of Na+ over here, but there's more potassium ions down over here, that actually maintains what we call membrane potential, and eventually it will actually send an electrical impulse to the cell next to it, which will send an impulse to the cell next to that and send the signals up to your brain. And so you need your certain ion levels, and we call these ion levels electrolytes because they have an electrical current. They have positive and negative charges. And so our electrolytes are really ion balance. The most important ones that we find in our body are sodium ions, potassium ions, calcium, magnesium, and chlorine ions that maintain our electrical currents across our cells. And so people always wonder, is it really better to drink these sports drinks after or during competition of a sports event? And the answer is absolutely yes, 100%. Water is not an ionic compound, which means it cannot replenish the lost electrolytes that you sweat out during your competition or during your exercise. So drinking sports drinks is usually going to replenish that and keep your electrolytes at a certain level. Let's talk about isotopes. Isotopes are different numbers of neutrons in the atom instead. So we're not talking about the electrons, we're talking about the neutrons. Anytime that we talk about the neutrons, we are talking about the mass of the atom. And it's not going to be the normal one that's on the periodic table. For instance, this is a normal lithium because most lithium on this planet has a mass of 7, which means that it's going to have 3 protons and 4 neutrons. But sometimes you could have lithium-6 or lithium-8. Lithium-6 is when there's 1 less neutron, so there's 3 of each protons and neutrons. But lithium-8 is when you have an extra neutron than the normal lithium on the periodic table. Your atomic mass is usually a decimal number, such as the sodium was 22.9 something. That's because it's an average of all the isotopes of that atom on the planet. For example, down here, we have carbon. The main carbon atom on this planet has a mass of 12, which means that there's 6 protons and 6 neutrons. But even though that this makes up approximately 99% of all the carbon on this planet, there's kind of a little less than a percent that actually is carbon-13. So we would write carbon-13 like this. Carbon-13 is an isotope of this one. That means it has more neutrons than normal. And then we have carbon-14, which you would write like this as an isotopic symbol. And carbon-14 is when there's even more neutrons, and so it increases its mass. Now, if you increase a mass too much or too little, the neutrons and the protons can start to react kind of adversely, and they're moving around trying to become stable because they're kind of unhappy. And as they're moving around trying to become more stable, they actually release a lot of energy and particles, and we call the energy and particle loss radiation or radioactivity. And so when we look at the human body, we could actually look at how radioisotopes or radioactive isotopes are important for us. And what do we use them for? Because most people know radiation is being harmful to you and could cause cancer. However, we use something called nuclear medicine to actually image body parts, detect and diagnose problems, and treat diseases. The picture down here is actually showing a PET scan. We're going to talk about PET scans, but it shows a PET scan, and the colorful areas showing in the spine and the bones, there's actually leukemia present in the bone marrow, and they use a radioisotope to tag it and detect whether or not there is leukemia. Over here, we could do that with the brain too. So they could do a PET scan of the brain to look at its activity, and they could see that, when somebody is intoxicated, there's a lot less activity in the vital parts of the brain than somebody who is not intoxicated. So they could look at that as well. And also, they could use it to treat diseases. Treating diseases, they actually use it during chemotherapy. So they use radioactive isotopes to help kill the cancerous cells. Radium and cobalt are 2 of the radioactive isotopes that they could use for that purpose. And even looking at your thyroid, if you're hypothyroidism or you have a goiter or something, your thyroid's all messed up, they will actually use radioactive iodine to figure out if there's a problem with your thyroid. So they use this all the time in nuclear medicines. Compounds versus molecules. A compound is simply different atoms that are combined to one another, and then a molecule could be either 2 of the same atoms or different atoms as well. And when we talk about the human body, we're focused more on the molecules because they're mainly covalently bonded. So let's look at how do you get these atoms together, and you do that by doing chemical reactions. A chemical reaction is pretty much when you break or make bonds between two or more different things to make new stuff. So the making and breaking of bonds. And here's a famous chemical reaction. This is cellular respiration. This is how we make our energy in our mitochondria of all of our cells, where we take our glucose sugar and we breathe in oxygen. We do some chemical reactions in the mitochondria and it breaks them apart. They find new partners to make ATP energy, which is our main source of energy. But we also have a couple waste products in the meantime, and that's how we do it. And so this shows a better picture of it. You take different molecules or compounds, and a chemical reaction will break them apart from their partners. They will find new stable partners, and through making and forming new bonds. Now there's different types of bonds that you can make. So let's look at the 4 different types of ways that you can connect atoms to atoms or molecules to molecules. The first one is an ionic bond, which forms when you make ions. Because the ions, as they gain or lose electrons, they become positively or negatively charged. So the positively or negatively charged atoms will be attracted to one another. And if you look at this example down here, we have sodium and chlorine. Sodium will give up its electron to chlorine to become stable. And so sodium gets a plus charge, chlorine gets a negative charge, and you get sodium chloride. That is an ionic bond because positive and negative will be attracted. Magnesium will lose their 2 electrons to oxygen, and so magnesium will get a plus 2 charge, and oxygen will get a -2, and you get magnesium oxide, which is an ionic bond, and then you get calcium chloride here when calcium gives an electron to each of these chlorines. And so an ionic bond is when the oppositely charged ions are attracted to one another. The next type of bond is the strongest type of bond that there is, called a covalent bond. And covalent bonds is when you share electrons with an atom. So you're not actually transferring electrons or gaining that electron, you share them within your orbits. And so water is a really good example of covalent bonds because the hydrogen needs 1 more to become a full valence shell, and the oxygen needs 2 more to have a full valence shell. So they will share their electrons with each other in their same orbits. And so this sharing is called a covalent bond. And you could see the sharing in each one of these examples between hydrogen, fluorine, oxygen, and carbon to make carbon dioxide. Here you could set, this would make a double bond. So a lot of times we write it like this when it's a single bond, and if it's a double bond and you actually draw it out, this would be carbon dioxide. And then this is carbon sharing with 4 fluorine atoms. So again, you are sharing your electrons. There's 2 types of covalent bonds. There's 2 ways that you could share. You could share equally or unequally. If you share equally, it's called nonpolar because they're not being pulled to the poles of the atom or the molecule. And so in this example, you notice that these electrons, which are X's, are in the middle of these 2 atoms in the yellow circles, and they're not being pulled one way or the other. So that's an equal sharing nonpolar covalent bond. But if one atom is stronger at pulling the electrons towards it and pulls it towards it more than the other one, then electrons are negative, so you're going to get those negative electrons over here. It's going to give one side a slight negative charge, which means that over here there's not really so many electrons, and so this one's going to have a slight positive end. And that's called a polar covalent bond because the electrons are pulled towards one pole. If you look at the hydrogens, they're equal strength, so they are going to be nonpolar. If you look at the chlorines, they're the same strength, so this is a nonpolar. But if you put a hydrogen with a chlorine, the chlorine is much stronger. We call it electronegativity, has a stronger electronegative pool, which means it loves to hoard electrons. And therefore, it's an unequal sharing, so we call this a polar covalent bond. So as a review, if you transfer electrons, you're called an ionic bond because the negative and positive unite. If you share electrons, you are covalent. If you share them equally, you are non-polar covalent. But if you share unequally, you're going to get a positive and negative partial side, which is called polar covalent. And then we have 2 more. Hydrogen bonds is when you get a positive hydrogen that's connected of a polar molecule that's connected to another part of a polar molecule. And water is the biggest one that we look at when we look at hydrogen bonding. So here's a water molecule. Water's polar, which means oxygen loves electrons, and so it will pull all the electrons down towards its end. These hydrogens in white they're not as strong, so they're not getting the electrons on their end, so it's going to have a slight positive charge. Well, this is a polar molecule. Polar molecules sort of act like little magnets. And so when it comes across another polar molecule, in this case another water molecule, the positive hydrogen, there's a negative oxygen and another positive hydrogen. Well, the positive hydrogen of one is going to be connected or attracted to the negative part of another molecule. And so this right here between molecules is a hydrogen bond. It's negative between the positive parts of pretty much polar molecules, hydrogen bonds. Your DNA, hopefully you remember that A's always pairs with T, and C's and G's always go together, and this bond that holds them together, which are shown right here and right here with these base pairs, that's a hydrogen bond. So that's the positive hydrogen that's being attracted to another negative part of a polar molecule. Our proteins in our body are formed, they have to fold together in these weird shapes. And so this is a folding structure of proteins in your body from simple to complex. And this secondary structure is actually this bending or this twirling nature. That's those bonds that make it bend or twist are actually hydrogen bonds. So they're important in living things, obviously. The last type are van der Waals forces. Van der Waals forces are not a true bond because you're not really transferring electrons, nor are you in hydrogen bonding. But these are very short-lived, quick attractions between molecules holding them together. So this is how the gecko walks up a vertical surface. It's not because it has sticky glue or suction cups. It's because the atoms that make up the millions of tiny microscopic hairs in its little fingers over here, the atoms that make it up are exhibiting van der Waals forces. Electrons are constantly in motion, and just by chance, all the electrons could end up on one side of an atom, and so it gets a slight negativity, and the other side is slightly positive. And so when negative and positive attract each other, you could stick to something. And so that's actually how van der Waals forces actually work. And when we look at our protein folding again, when we get up into the third structure, the tertiary structure of protein folding, a lot of the forces that hold this weird shape together in a protein is done by van der Waals forces for some of them. And so those are the different types of bonds and a quick review of chemistry. Hopefully that sparked a few light bulbs and you're like, oh yeah, I remember that, and I hope it was helpful.